So, good evening, everybody. My name is Professor Paul Townsend. I'm Pro Vice-Chancellor and Executive Dean of the Faculty of Health and Medical Sciences here at the University. And this is a very, very special evening, and I'm privileged to be your Master of Ceremonies, which means I have to be well behaved and monitor your questions. So I'd like to thank you all for attending what promises to be an enlightening and I'm sure insightful lecture delivered by tonight's guest speaker, very, very well celebrated Professor Dame Sarah Gilbert. Sarah's contribution to the field of vaccinology is incredibly inspiring, integral to the development and testing of the universal flu vaccine, lead developer of the Oxford AstraZeneca COVID-19 vaccine, made a dame in the 2021 birthday honours for services to science and to the public health, and only last week became a member of, or a fellow of the Royal Society, FRS. It is estimated that within the first year of the Oxford vaccine being available, that over 2.5 billion doses were administered across the world. That is a phenomenal number. No wonder then that she received a standing ovation at the Wimbledon Tennis Championship in 2021. But more importantly, had a Barbie doll made in her honour. So Sarah's success the year before as the Oxford project leader for the vaccine against the novel coronavirus SARS-CoV-2 owed much to the dedication and perseverance earlier in her career. It was a career that almost didn't happen, as after graduating from the University of East Anglia with a degree in biological sciences, she considered leaving science while working on her PhD, somewhat disillusioned by the lack of interaction with scientists from other disciplines. But thankfully, and the world is very thankful, she stuck with it and found a job with the Brewing Research Foundation here in Surrey, where she worked on aspects of yeast genetics and biochemistry. So after tonight's lecture, you may wish to raise a glass to them. Right. By her own admission, Sarah's transition to human health was slow, but her rise since then has been anything but. And her passion for developing viral vectored vaccines remains undimmed. That accelerated pace of change is reflected in the way that she has helped to transform vaccine delivery and the manufacturing of those vaccines. A proud advocate for the value of interdisciplinary research, her commitment to collaboration and teamwork can only be admired. It now gives me great pleasure to hand over to our President and Vice-Chancellor, Professor Max Lu, to formally introduce Sarah. Thank you.
Good evening, everyone. On behalf of the university, I'm so delighted to welcome you all on campus in person and also on the live stream. Of course, especially our honored guest speaker, Professor Dame Sarah Gilbert, and other distinguished guests to this year's Adam Sweeting Lecture. This lecture series, some of you been here before, has a proud history of hearing from eminent speakers at the forefront of science and technology. This lecture bears the name of two of Surrey's pioneers, Professor Alf Adams and Professor Martin Sweeting. I'm pleased to see both of them present tonight. Like Sarah, they both push the boundaries of knowledge through inquiries and discoveries and to innovation, translating their ideas into very impactful outcomes that have changed the world. For example, Professor Alf Adams, by inventing the strand layer quantum well laser that actually laid the foundation for the internet streaming, and Professor Martin Sweeting for pioneering the modern small satellite that has given us the great space industry that we now globally. The impact of their world-changing discovery inventions is simply huge. Besides the inventions of solid-state laser and small satellite here at Surrey, we also take pride at our great record of many other scientists, engineers, and researchers that delivers real-world impact. And tonight, we are so privileged to be in the company of an exceptional scientist, as we just heard from Paul's introduction, who has just not just made an incredible impact in her own field, but indeed extraordinary impact on humanity. Dame Sarah Gilbert's impact in vaccine development is felt literally by millions around the world. I'm not just talking about the sharp needles. Rather, as a result of her dedication and perseverance in the field of vaccinology, millions of lives have been saved and protected. So we're very grateful to her work and her colleagues' work as a team. And Sarah would be the first to acknowledge that these achievements are not hers and hers alone. A very large team was, of course, involved in the development of COVID-19 vaccine. And importantly, this was the latest step in over 25 years' journey in this field. Day in, day out, Sarah and her colleagues strove to develop the Oxford vaccine once the sequence was released from China in early 2020. And despite working in very trying circumstances during the lockdown, I mean, I heard the stories about your team were virtually starving for days and really didn't even get the food. Her team, and drawing on what has been learned from the development of the MERS virus, had preclinical results in a matter of weeks. It takes a huge commitment, ladies and gentlemen, to achieve something so significant so quickly, and it illustrated the integral part of Sarah's ethos and her leadership and her collaboration in her research and development, not least working closely with scientists and engineers from AstraZeneca. We here at Surrey do recognize the importance of interdisciplinary and cross-academia to industry collaborations and partnerships in order to solve the world's more pressing challenges, such as climate change, global health, of course, and many, many other challenges we're facing today. I also think that it is critical to work with practitioners and policymakers in order to make a difference to society. And therefore, social sciences and humanities are as important. I think Sarah does embrace this broad view, and she believes that discoveries are often made at the boundaries between disciplines, and that the success in developing a vaccine, for example, so quickly was due to the effective research, testing, and manufacturing activities in parallel and in a very coordinated fashion. To integrate all this team's effort into productive outcomes, as we saw as the AstraZeneca Oxford vaccine, required tremendous leadership, and that strong leadership provided, and also the resilience by Sarah to the whole project was so instrumental and so inspiring. So it is one of the traits which, as fellow scientist, I admire most about her. I would like to take this opportunity, of course, now to salute Sarah for her dedicated and pioneering research and innovation that has transformed lives around the world. Now, Sarah, it gives great pleasure to invite you to give the Adam Slayton lecture. Please, welcome.
Thank you. Well, it's lovely to be here this evening. See so many of you at in-person events again. It is really wonderful. I've been going to quite a lot of conferences around the world recently, but not so many events with so many members of the public as we have here today. So what I would really like now is my slides. There we go. Okay. So I've got a bit of a mixture of slides for you. Some of them are more technical than others. Don't worry, it doesn't get progressively more technical. I'll try and point out the salient points of the more technical ones and talk you through the rest. So the work that we did in 2020 obviously didn't start out of nowhere. We didn't just suddenly decide on the 1st of January 2020 that this would be a good field to get into. Many of my colleagues have been working with me in this field for quite a long time previously, doing technology development, working on vaccine manufacturing, establishing a setup on one campus at the University of Oxford, on one hospital site, where we could do the early design, the thinking about vaccines, the early design, the early production of vaccines for preclinical testing, and then go straight into a GMP manufacturing facility. That's a special, very high quality manufacturing facility that we use when we want to make vaccines to immunize people in clinical trials. We don't just take something out of the lab and put it straight into the manufacturing facility. There has to be a very careful process that we go through before we can even start. And then it's the facilities are licensed by the Medicines and Healthcare Regulatory Agency. Everything has to be approved. The actual batch of vaccine we make has to be approved once we've finished making it and testing it. But we can do that in a university facility, which is right next to our clinical centre, where we can then vaccinate people, having received all the approvals for the ethics of the trial and the regulatory status of the vaccine. We can then take the vaccine across the road, start vaccinating people, take blood samples from them, and then take those back to our research labs and start looking at the immune responses in those blood samples. So we'd already got all that set up. We've been doing it for quite some time. What we had to do in 2020 was different, firstly because we went so much quicker than we ever had done before, and we weren't used to joining up the different pieces of vaccine development very quickly, because the way research normally works is you're always waiting for the money. So we might manage to raise a little bit of money to get started, generate a small amount of data, then write another grant application, try to get the money raised to do the GMP manufacturing and the first clinical trial, and so on. So the reason that vaccine development has traditionally taken a long time is largely because we just don't have the money to go any faster. So there's a lot of gaps that we can close up if we're not waiting for the money each time. But we hadn't actually planned ever how we would do this if we wanted to go really quickly. So we had to kind of work all that out on the hoof and see how we were going to do it. And the other problem was that by the time we actually started our clinical trials, we were in lockdown. So we'd always thought of ourselves as manufacturing a vaccine for a virus that was causing an outbreak, doing a clinical trial in volunteers, healthy volunteers in Oxford, maybe a few other UK sites, and then providing the vaccine and all the information about the vaccine to wherever in the world the outbreak was happening. But of course, that's not the situation we were in by April of 2020, which is when our clinical trial started. We were in lockdown. So we had to work out how we do clinical trials where you actually need to talk to volunteers and immunise volunteers and take blood samples from volunteers while we're in lockdown, complying with all the social distancing regulations, and also keeping our teams socially distanced when they were working in the research labs. Wasn't too difficult to do because the campus closed down apart from work that was recognised as essential COVID work, and so we were able to spread out and people had a lot more space to work in than they normally would do, which was very beneficial for us. But then we didn't have enough people, so we had to start calling in more people that normally work on other projects but have the right skill sets so that we could add to our team and accelerate the work that we were trying to do. So it was challenging, but we continued to identify the problem and find the solutions until we got through the vaccine development in 2020. But I want to go back now to 2014 and think about an earlier outbreak of a different virus, Ebola virus. Now, in 2014, if you remember, there was quite a large outbreak of Ebola virus in West Africa in 3 different countries, Liberia, Sierra Leone and Guinea. And this wasn't the first time that this virus had been identified. It was first identified in 1976, causing cases in Central and West Africa. And there would normally be a small outbreak about every year since 1976. But it didn't spread very quickly. And because the outbreaks tended to happen in rural areas, it's a disease that's carried in bats, and if people are in contact with infected bats, they can get infected as well. That doesn't really tend to happen in cities. It's in the more rural areas where there are more space between people, and so you don't get such rapid spread of the virus in that kind of circumstance. Once it was recognised that there was an Ebola outbreak, and it's a hemorrhagic disease, it causes a lot of bleeding, roughly 50% fatality rate, and quite easily transmissible between people if they're in close contact. But once an outbreak was recognised, then the standard public health measures, contact tracing, quarantine, getting people to stay isolated away from other people until the outbreak ended, those had always worked. But in 2014, what was different, the virus got into cities, it started to spread very much more rapidly, and we started to see this epicurve with having rumbled on all the way through the early part of the year. It started at the end of December 2013, rumbled slowly on mainly in Guinea, and then suddenly picked up and started to spread very rapidly, and the world had to start to take notice. And there were lots of interventions that were planned, lots of field hospitals set up, but it was also thought that we should be testing a vaccine. And nobody had been trying to develop a vaccine against Ebola to use in this kind of situation. There were some vaccines that had been through early stage development, mainly in the US, which were designed to use if somebody wanted to use Ebola as a bioterrorist weapon. So it never thought about how they would actually be used in an outbreak area. Sorry, I'm trying to move the slides on. I'm not getting any success. Okay, so we were asked if we could help trial a vaccine that had been part way through development. It had been manufactured, it hadn't been put into vials, clinical trials hadn't started. It was an adenovirus-vectored vaccine. It was based on a particular adenovirus called ChAd3. And the only other clinical trial of a ChAd3 vectored vaccine that had taken place anywhere in the world had happened at our clinical unit in Oxford, with the MHRA as the regulatory authority. So it was felt that in this urgent situation, the fastest way to do a first-in-human clinical trial, test this vaccine in people for the first time and look at the immune response, would be to ask us to do it in Oxford with MHRA approval. And so the Wellcome Trust funded it very quickly. We got the vaccine put into vials. We got all the last bits of testing done once it's gone into the vials. We worked closely with the MHRA to get a trial submission to them, and then very rapidly got approval, both regulatory and ethics approval. We got the vaccine shipped, we got the vaccine labelled, and vaccine labelling is a really big deal. It's not just sticking a label on the vial. There is an awful lot to labelling a vaccine for a clinical trial, with very many approval processes that you have to go before you can put that label on the vial. Got that done, we got the trial contract signed, and in the middle of September, so a month after starting to be involved in this process, we'd vaccinated our first volunteer, and a month later we'd vaccinated the 60th, and we were able to provide information about the reactogenicity and the immunogenicity of this vaccine to a group in Mali, where they took the vaccine and they started testing it in Mali. This is a country next to the outbreak area. It was tested in Malian healthcare workers who were likely to have to go and respond to the outbreak if it spread into their country. And that was done in another month. So by the end of December, we were expecting to see this vaccine going into trials in the outbreak area, so in the 3 countries where there were most of the cases, but that didn't happen. We had to pass on our data. It was up to somebody else to do it at that point. And what actually happened was there was a pause while there was a lot of debate about the ethical way to do clinical trials of a vaccine to see if it works when the disease has a 50% fatality rate. And those discussions went on for some time. Now, those discussions could have been held ahead of time, but they weren't. And so it wasn't until April of 2015 that a vaccine went into efficacy testing. By that time, the numbers of cases had dropped dramatically due to the public health measures, nothing to do with vaccination. So by the time anybody was ready to test the vaccine in April of 2015, there were very few cases left. We should have been testing the vaccine at the peak of the outbreak here and here, but no, we were in this situation and there wasn't very much infection, which was obviously great to see the outbreak was being brought under control, but we missed the opportunity to test this vaccine. So only one vaccine went into efficacy testing, and it wasn't the one that we'd helped develop. It was one from Merck, another viral vector vaccine, somewhat different characteristics. And fortunately, it turned out to have really high efficacy, which was great. What wasn't so great was it needs ultra-low storage temperatures, -80 freezers, to store it in and transport it in, which isn't ideal for Africa. And also the manufacturing process wasn't really scaled up, so it's quite difficult to manufacture large quantities of the vaccine because the development work hadn't been done before this efficacy trial was run. So it's good news that there was a vaccine. We didn't get to find out if the vaccine that we'd helped to work on would have been effective, although looking at the immunology results from the VSV vector vaccine and the ChAd3 vector vaccine after the event, you would have expected that that would also have been highly effective, but we never got the chance to find out. Another vaccine from Johnson & Johnson was licensed later on. That was done by comparing immune responses to the level of immunity that you need to protect a monkey who's then deliberately exposed to Ebola virus. So we felt that we'd done a great job getting things off to a quick start, but then everything stalled, and then actually we didn't get the vaccine efficacy testing that we'd expected to see and that we were trying to help accelerate. So that gave us the idea that if we're going to do this again, we really want to try and think through the whole process, not just think about phase 1 trials, but think about how do we go from phase 1 to phase 2 to phase 3? So phase 1 is a small group of people, healthy young adults, looking at how they react to the vaccine and what immune responses they make. Phase 2, you expand the age range. You would start to include people with comorbidities, people with some health problems that are stable, and start to look at how the vaccine behaves in that population. And then phase 3, you test to see if the vaccine is actually effective against the disease. And that's where you need tens of thousands of people typically to find out if the vaccine actually works. So the technology that we've been developing was very similar to that ChAd3 vectored Ebola vaccine. It's another replication-deficient simian adenovirus. So let me talk you through what that is. So this is a diagram of an adenovirus. It's a fairly small and simple virus which has a very rigid structure, and it has these spikes with the knob protein on the end that allow it to stick onto a cell. Once it attaches to a cell, it will then get inside a cell, and when it does that, the DNA of the adenovirus will go into the nucleus of the cell, and that will take over, and it will start to make more copies of all the adenoviral proteins, which then assemble around the extra copies of DNA that's been made inside the cell, and the cell becomes a factory for making more adenoviruses, and then they burst out and they go off and in fact lots more cells in your body. So adenoviruses normally cause respiratory infections, fairly mild respiratory infections. They can cause infections in the eye or gut disease as well. Not severe disease. But we get infected with adenoviruses all the time. If you have a cold, it could be an adenovirus. We don't diagnose it. It's not a serious illness. But we can use these to make a vaccine. So what we do is we don't want the virus to be spreading through the body when we're using it as a vaccine. So we take out some of the genes, it's the E1 and also the E3 gene, that mean once this vaccine, once this attaches to your cell and goes inside it, and the DNA goes into the nucleus and starts to make the protein that's encoded on that DNA, then it can't go any further because the DNA can't make more copies of itself, and so you can't make any more copies of the virus, and it doesn't spread through the body. So it's safe to use and it's controlled. We put it in, infects the first cell it finds, it makes the protein, because what we've also done is added a gene from a different virus, and the vaccine then produces protein that's encoded by that gene, and that's the protein that we get the immune response to. So most of the protein that the adenovirus is now telling the cell in the body to make is, in this case, the spike protein of the SARS-CoV-2 coronavirus, and that's why we get a very strong immune response against that, because the body recognizes it's something new. The immune system has to distinguish between self and non-self, friend and foe, and if there's something new there that the immune system hasn't seen before, it will start to react against it, make antibodies, make a T-cell response, but in a safe environment, because this adenovirus vectored vaccine can't make you ill, it doesn't spread through the body. But then you have that immune response, you have the immune memory, and then when you do encounter the SARS-CoV-2 virus for real, you've already got the antibodies, you've got the T-cells ready to respond quickly. And even if it doesn't stop you getting infected, it stops you getting seriously ill and it stops you dying. So one of the reasons we use this type of technology to make vaccines is it's what's called a platform technology. We can use the same technology to make vaccines against lots of different diseases simply by changing the gene that we add into the adenovirus. Huge advantage of that is that we can increase our understanding of the technology before we use it to make all the different vaccines that we want to make. We spend our time working on the technology, how to manufacture these adenovirus vectored vaccines, how to store them, how to test them, what dose should we give to people, what's their likely response to the adenovirus vectored vaccine. And then because we've done so much work on the technology, when we come to develop a vaccine against a new virus, there's only the last piece of the work to do to test the new vaccine against that new virus. So this means we can go quickly, it's much more predictable. We're not trying to start from zero and make a vaccine against a new virus. We've already done 95% of the work, we just need to do the last bit. And we'd been working on this for some time. We'd made the adenovirus into about 10 different vaccines. Up until that point, we'd done 12 phase 1 studies. 330 people have been vaccinated with ChAdOx1 vectored vaccines in clinical trials, largely in Oxford, but also in some other countries as well. And ChAds more generally, so simian adenoviruses apart from ChAdOx1, like ChAd3 and ChAd63 and some others, overall, about 6,000 people of many different ages, going from babies in Africa, in malaria vaccine trials, through to older adults in the UK in flu vaccine trials. So been tested in a wide range of people. And we can see a consistent safety profile and also strong immune responses even after one dose. And if you want to try to control an outbreak, you really do want strong responses after one dose. So some of the more old-fashioned ways of making vaccines will not give you a good response until you've had 2 doses. So that's not ideal when you're trying to control an outbreak quickly. This will give you a strong response very quickly. And we'd also been working on another coronavirus, the Middle East Respiratory Syndrome. And we'd shown that when we vaccinate monkeys with the adenovirus expressing the MERS spike protein, they made immune responses, and when they were exposed to MERS coronavirus, they didn't become ill. And also we've done phase 1 trials of the MERS coronavirus vaccine in the UK and in Saudi Arabia, which is where most cases of MERS occur in the world. That had been done prior to 2020. So we've done a lot of work on developing the technology and some work on developing a coronavirus vaccine using this technology. So some of the other uses that we've been putting it to are to develop vaccines against other outbreak pathogens. So Nipah virus is one that's found mainly in Bangladesh, although first identified in Malaysia. It's carried in these fruit bats. They're not affected by it. They can be infected with Nipah and be perfectly well and still fly around and eat fruit or get into pots that people are using to collect date palm sap, and they contaminate it with the Nipah virus if the bat happens to be infected, and then people can get infected. Or also happened in Malaysia, pigs got infected by eating contaminated fruit, and then people got infected from the pigs. Most of the cases are in Bangladesh, and the case fatality rate, so that's the percentage of people who die if they get the infection, is anything up to 75%. And these fruit bats travel long distances, so it's quite hard to know exactly where these infections are going to crop up. There aren't any vaccines against Nipah virus, but there's a related disease. I don't know where I'm supposed to be pointing this to make it work. There we go. There's a related virus called Hendra virus, which is a close cousin of Nipah virus, and this infects horses, and it was found infecting racehorses in Australia, and then people were getting infected from exposure to the infected racehorses. And again, bats form the reservoir. But there is a vaccine against Hendra virus, which is fairly simple technology. It's recombinant protein delivered with adjuvant, and this has been very effective against protecting the horses against Hendra virus, and that means that people don't get infected because they're no longer being exposed to the infected horses. So this illustrates the One Health concept in vaccinology. Sometimes we're better off vaccinating the livestock that are getting infected that the people are then interacting with. Sometimes we can't do that because the reservoir is a bat. But if it's a pig or a horse or a sheep or a goat, sometimes the best thing to do is to try to develop a vaccine against the livestock species. But if they don't make the livestock species ill, which is often the case, then it's very difficult to find the money to persuade anybody that you should be vaccinating those livestock to protect human lives. But the fact that there is a vaccine against Hendra virus means there's very good prospects of having one against Nipah virus as well. Another virus for which we don't have any vaccine, well, there's only one vaccine of a vaguely related virus out of a very large group, is Lassa fever virus, of which 200 million people across West Africa are at risk of infections. It's carried in these small rodents, the multi-mammet mouse. They like to get into people's houses and they'll contaminate food stores, and people can get infected in that way. It's very, very dangerous in pregnant women. It's dangerous for the healthcare workers who are looking after those pregnant women. And in children, although it doesn't necessarily cause a severe disease, it results in permanent deafness in about 10% of the people who get infected, even though they only have a mild illness. About 20% will be deaf after a Lassa infection, half will be permanent and half will be temporary. So there's a lot of deafness that's caused by this disease, as well as illness and in some cases deaths in pregnant women. And we have no vaccines, but we've been starting to use Chalax-1 to develop a vaccine against Lassa fever, and also starting to use the Chalax-1 technology to make a vaccine against MERS coronavirus. Can I have the next slide, please? Okay, so MERS was originally also probably found in bats, but then camels got contaminated, got infected, and camels in Africa traded into the Middle East have then spread the virus between them. It gives camels a cold. They're not really ill. The majority of camels in the Middle East have now been infected with MERS coronavirus at some point, and at the time that they have an active infection and they're shedding virus, which these camels don't, they don't have runny noses. But if they are shedding MERS coronavirus, then people can get infected. And for older people, maybe with compromised immune systems, it can be a severe or fatal illness. We now know that, as with SARS-CoV-2, young, healthy people can have a really very mild illness with MERS coronavirus, but they can also then transmit it to other people. So it sometimes gets transmitted through people who work with camels to older members of their family who then become ill. If it gets into a hospital where there are a lot of people with compromised immune systems, it spreads very, very fast. And there was a big outbreak in South Korea where one businessman went to the Middle East, got infected, went back to South Korea, visited 3 different hospitals, and there was a large outbreak. I think nearly 100 deaths, and it wiped billions of dollars off the Korean economy in the aim of trying to control this outbreak. They had to lock down and shut everything down until it was all completely sorted out. And that was a result of one person travelling back to their home, having been travelling in the Middle East and getting infected with this virus. So we really need a vaccine against MERS coronavirus. And as I said, we had been starting to work on this prior to 2020. So we had some data, some clinical trial data. Any thoughts?
No idea.
Okay, so this is what the kind of data that we collect from the blood samples of our volunteers. So I'll try and break this down for you a bit. This is a time course. Day 0 is the day the person gets the vaccination. And these are antibody responses against the MERS spike protein. Now, they're not completely negative on day 0. That's because there are 4 seasonal coronaviruses that circulate and cause us a cold, and we don't worry about them very much. But there's a bit of cross-reactivity in the antibodies between those coronaviruses and MERS coronaviruses. So depending on when people last had a cold that was caused by a coronavirus, they'll have a bit of an immune response here. But 28 days after we give them a single dose of our Chalex MERS vaccine, we see a big increase in antibody responses. And although these decline somewhat, they then plateau and they're still present a year later. Now, this is absolutely normal for an antibody response after a vaccination or infection. And there's been a lot of talk in the press about waning immunity and we've lost immunity to SARS-CoV-2. This is what happens. This is what should happen, but the immunity doesn't completely go away. We don't lose it altogether. We also look at T-cell responses. So here we've got the different dose groups divided. The higher dose of vaccine gives you the highest immune response. Here they're all mixed up together. An increase in T-cell responses, the other main part of the immune system, again increases after vaccination and declines somewhat, but is still maintained a year after vaccination. And we look at neutralizing antibodies as well. So these are the antibodies that can bind onto the virus when it starts to invade the body and stop them getting inside our cells. That's not the only thing antibodies can do, but this is a very rapid way of getting protection against infection if we have a good neutralizing antibody response. And we saw that best at the highest dose that we gave in the MERS trials, and that ended up being the dose that we then used in the SARS-2 coronavirus trials.
Could you please point it to the big screen?
Point it to the...
Over there. There you go.
Okay, thank you. So this is the timeline of the Chalex 1 vaccine development starting in January 2020. We had our GMP manufacturing started in February. Our first clinical trial started in April. Unlike the Ebola virus trials that I told you about, we were planning all the way through to phase 3 and testing efficacy this time. So our first trial started in April, and our second trial started in May, and our third trial started in June. And these were getting bigger and bigger and bigger as we realized what we were going to need to do to this. And we started trials in not only in the UK, but in Brazil and in South Africa. And AstraZeneca joined us and they took over manufacturing, and they started phase 3 trials in the USA and Japan and India. We had lots of people immunized, and then we have to wait to see. We have to wait for people to get infected, because you can't tell if the vaccine works unless people get infected, and you just have to wait for that to happen. Then you compare the number of people who had the placebo or the control vaccine to the number of people who had your new SARS-CoV-2 vaccine, and that tells you how effective the vaccine is, or it gives you an estimate of it. It's important to remember that you can't really compare these estimates of efficacy between different trials because they're done in different circumstances in different populations. But when we got our efficacy data, we were able to see that across the whole trial, we were seeing 70% protection against infection. This was symptomatic infection, not severe disease, in our trials. But it wasn't consistent across the different groups. There was one group that had a 90% efficacy, and some only had 63%. So why was that? Well, it turned out to be that the people who had the highest level of efficacy had the longest interval between the 2 doses of vaccine that they got. We discovered that giving a second dose would increase the immune response. We don't know how big the immune response needed to be to protect against this virus, so we stuck with giving 2 doses. But because we were trying to ramp up manufacturing as we expanded the clinical trials, we weren't able to give everybody the same dose 4 weeks apart. So some people had to wait longer between their doses. That turned out to be a really useful piece of information to have, because the longer the interval between the 2 doses, the higher the vaccine efficacy. And this is because the longer the interval between the 2 doses, the higher the immune response. So if you wait 3 months between the first and the second dose instead of 1 month, you get a stronger immune response and you get better efficacy. And that then fed into the UK approval, which was to authorise this vaccine for 2 doses with a 3-month interval. And this was very useful in another way as well, because at the time we started COVID vaccinations, December and then increasing in January of 2021, there was not enough vaccine available to vaccinate everybody. Manufacturing was catching up, but there was quite a lot of catching up to do. And we didn't have all the vaccine that we would have liked to have available. So by giving people their first dose and then asking them to come back 3 months later for their second, it meant that more people could get their first dose more quickly and be protected. But what about the people who've only had 1 dose? Are they suffering? Are they losing out by having to wait 3 months till they get the second dose? And the answer is no. Efficacy after the first dose is above 70%, and up to 90 days, that doesn't decrease. And it's planned to get the second dose at 84 days. So once you've had your first dose, you've got a good high level of protection. As I said before, that's what we want from an outbreak vaccine, good protection after the first dose. If you wait 3 months to get the second dose, you increase the protection, gets better, it will last longer. And so that's how the vaccine rollout was done. And it was also the approach that was taken for the RNA vaccines. Even though there wasn't actually any data on different dose intervals for the RNA vaccines, they were also given at a 3-month interval. So in clinical trials, we're testing to see, is this vaccine working? Can we actually start to use this to protect the whole population? The exact level of efficacy that you read out from those clinical trials will depend very strongly on what you're trying to measure, what you said the clinical trial endpoint will be. If it's symptomatic illness, so mild illness, PCR positive, this is what we used in our trials, and we got an efficacy overall of 70.4%. We also had people send in nasal swabs every week, even though they didn't have any symptoms, and tested those, and some of those turned out to be positive, even though people were perfectly well. So it turns out against asymptomatic infection, we've got 27% efficacy, so much lower than protection against mild illness. Against severe disease, hospitalization, 100% protection, and against death, 100%. That doesn't mean that when these vaccines are used in the general population, we will always see 100% protection against severe disease and death, because this is in a cohort of 11,500 participants who may have had stable comorbidities, but they weren't people who are as ill as some of the people who are going to receive the vaccine are. So depending on what you're defining protection against, you can get a reading of anything from 27% up to 100%. And if you focus in on this area, which is what was used as the endpoint in clinical trials, if you have a slightly more moderate disease definition than a mild disease, you're going more to this point. So the exact definition of that mild disease really matters if you're going to compare readouts from different trials. What we really want to know, as I said, is the vaccine working? Is it safe? Is it effective? Should we be using this in the population? And then what we want to know about is vaccine effectiveness, which is a different thing. This is, does it actually protect people when used for real? And we were very lucky to start to get data on that from Public Health England. On the 1st of March 2021, they released this data set showing that in England, people over the age of 70 who received their first dose of the vaccine, just the first dose, from 28 days after that, were at least 60% protected against symptomatic PCR positive disease, so feeling ill and getting a positive PCR test, 60% protection for both the AstraZeneca vaccine and also the Pfizer vaccine, and there was nothing to tell between them in this data set. In the over 80s, hospitalisation was reduced by 80% by this time, again with both vaccines. And what you can see on this graph is that deaths in the over 80s are reducing much more rapidly in the vaccinated population than deaths in those under 80 who had not yet been vaccinated. So already we're seeing a big impact on the reduction of deaths in the vaccinated population, even the oldest members of the population. And this then translated into the younger members of the population as we started to get down through those age groups. That translated into protection. And the picture of SARS-CoV-2 patients in hospitals completely changed after this point. They went from those terrible scenes of lots of people being sedated and needing respiratory assistance to actually people looking much more like a normal flu season. Because even if people do get ill after they've been vaccinated, the illness tends to be very much milder. So AstraZeneca were planning ahead. They were planning for manufacturing to supply as much of the world as possible, and to do this, they set up manufacturing facilities in 15 different countries around the world, trying to aim for local production, for local supply. And this is what resulted in 3 billion doses being released for use by the beginning of 2022 and an estimated 6 million lives being saved. This is because AstraZeneca set up this manufacturing network so that they could supply the vaccine as rapidly as possible. It wasn't as rapidly as anybody would have liked, but it was as rapidly as it could be done to supply the vaccine in very large quantities. And because this vaccine can be stored in a fridge, it doesn't need to be frozen to be transported, this makes it much more suitable for use in countries where they're used to using vaccines that get stored in a fridge and have facilities in place for that. But if you give them a vaccine that needs ultra-low temperature freezer storage, they don't have the facilities and they can't transport them and they can't use them. So this vaccine was very good for use in low and middle income countries. And the COVAX program was used to support global access equitably and get as many doses to as many countries as possible. It wasn't as fast as anybody would have liked, and part of the reason is that in large parts of the world there were no suitable manufacturing facilities, and it wasn't for want of looking, but there just wasn't anywhere that was ready to very quickly take on manufacturing of this vaccine. So there were 25 manufacturers that did do it, but we would have liked to have some in Africa and more in South America. That wasn't possible. And now there's a big initiative led by the WHO and the World Bank to set up vaccine manufacturing facilities in more countries, 5 new ones in Africa, for example, 15 new ones around the world, so that if we find ourselves in this situation again, it will be possible to start manufacturing in more places at the same time. We've shown that it's possible for one company to produce the seed materials and send them out to 25 different manufacturers who all then make the same vaccine. The regulators recognize it as the same vaccine, and that's very important. And that is how you increase vaccine supply. So if this all works, the next time there's a pandemic, there'll be more manufacturers, and Africa will get its supply from its own local manufacturers at the same time that Europe's getting its supply from its local manufacturers. So we did a lot of work on pandemic responsiveness in Oxford, starting from 2020. I've been talking about the vaccine development, building on all of the work that we'd done prior to 2020, and then bringing big teams together, partnering with AstraZeneca, thinking through as far as we possibly could at every stage. We changed our plans probably more than once a day for many months as we expanded. We brought in mobile units because we didn't have enough space to do our clinical trials. We had people bringing in food for people because there wasn't possibility to go out and buy it at the supermarkets. We moved people into a different accommodation because some of our staff were sharing accommodation with hospital workers and were therefore at higher risk of COVID infection themselves. And if one team member in the immunology lab had got a COVID infection, then the whole team would have had to quarantine. We would have had to shut down the immunology testing, and we didn't want to do that. So there was a lot of logistical planning that we weren't expecting ever to have to do. But we did it, and it all worked, and we didn't have any COVID infections in the immunology team or the clinical trial team. But that wasn't the only thing that Oxford did in the pandemic. Peter Horby, Professor Sir Peter Horby, as he is now, led the recovery trial. So this was a very large trial in the UK, which actually recruited 10% of the patients hospitalised with COVID, and in a very simple way tested drugs to see if they actually were of any use, if there were any benefit to people who were in hospital with COVID. Very, very quickly found that dexamethasone, a cheap and extremely widely available steroid, is in every hospital pharmacy around the world, and it doesn't cost very much. If somebody is hospitalised and requires oxygen support due to a SARS-CoV-2 infection, dexamethasone is a benefit. It's not a benefit if they don't require oxygen. So it shouldn't be given too early, but when they get to the point of needing oxygen, it's very beneficial, and that saved millions of lives. That data generated from the trial. They went on to test 11 drugs in total. They found 4 that worked and 7 that didn't, and it's very important to know about the ones that don't work as well as the ones that do. Hydroxychloroquine doesn't work, and you need to know that, because then everybody can stop using it instead of hoping it works or thinking it works, or would it work if you gave a higher dose? It doesn't, so don't use it. Use one of the drugs that do work, and that's what this trial did. And very few trials around the world actually managed to generate any useful information. Many trials were small trials within one hospital. They didn't have many patients in the study. They made it very complicated, and they didn't actually generate any useful information. The recovery trial was simple, it was large, it was rapid, and it generated very useful information. And this is a model that's being rolled out to study other diseases, because you need large numbers of patients all being given the same drug to find out if it works or not. And if you do that, you can find out very quickly, and you can then use that information to either treat patients with the drug or decide not to treat them with the drug. We also had partnership with different types of organisations, lots of UK organisations we worked with, lots of clinical trial sites around the world, and we also worked with industry. We had other people in Oxford working on diagnostics, testing the lateral flow test. There are many different brands. Some of them were great, many of them weren't. That testing was done in Oxford as well to find out which were the ones that we should all be using. And together we wanted to keep this team of people together, thinking across the board about pandemic preparedness and response. And so that's why we have the new Pandemic Sciences Institute, from people who worked on pandemic response and will continue to work together. Because if we work together, we can be much more efficient in the way we do things. We can have teams of people working on all aspects of Nipah virus response. So I'm working on a vaccine, some people are working on clinical trials, some people are working on surveillance, some people will make monoclonal antibodies out of the blood cells that we collect in the clinical trials that my team will run, and those can be used as therapeutics, and they can be used to develop diagnostics. And by bringing everything together, we can achieve more quickly, and be more cost effective in the work that we do. We will also focus on equitable access for everything that we develop. So I don't want to scare you, but there are quite a few other outbreak pathogens out there. So I've told you a bit about MERS in Saudi Arabia, Ebola in Africa, Lassa fever in Nigeria, Nipah originally, this is where they were first identified, SARS in China, of course, the original SARS, and then SARS-2. Rift Valley fever virus was first isolated in Kenya. It affects sheep, cattle, goats, camels. Camels can travel a long way while they're infected with Rift Valley fever virus and start infecting people in another part of the country or even in another country. Now, the whole of cases have happened in the whole of Africa. People get infected with it as well from their livestock, and it's another highly fatal hemorrhagic disease, and we don't have a vaccine. And this is one where actually vaccinating sheep would be hugely beneficial to stop people getting exposed to the virus. It also has economic benefits for the farmer, because of sheep infected with the Rift Valley fever virus, it's like it will lose its lamb if it's pregnant, and so it has big economic consequences for the farmer. Chikungunya virus is a very unpleasant infection where it causes really severe joint pain, long-lasting effects after infections. And you'll have heard about Zika virus, and there's recently been a Marburg outbreak. There's Crimean Congo hemorrhagic fever is another one that affects sheep and goats, and it's a hemorrhagic disease that can affect humans as well. And this is just the tip of the iceberg. What we need to be doing is developing the vaccines against all of these quickly and getting them stockpiled, getting them in the freezer. So as soon as we know that there's an outbreak, we've got a vaccine ready to take to that outbreak area and start using it to protect people, to protect the healthcare workers. We need to protect the healthcare workers for two reasons. Firstly, because they deserve to be protected, but secondly, because if they're not protected, they can become infected and they actually spread the disease amongst other people, against other parts of the hospital sometimes, or against a clinical unit when there is an outbreak. So we really have to protect them as quickly as possible, and we also need to protect the people when there's an outbreak happening. And we have the technology to make vaccines against these diseases. And these are not particularly difficult diseases to make vaccines for. There are some diseases like malaria, HIV, TB, where vaccine development is really complicated and very difficult. These are not difficult diseases to develop vaccines for. We just need the funding, and then we just need to do it. So the normal development timelines are about 5 to 10 years, doing everything in sequence like this. But as I said, there's often big gaps between these while we just wait for money. What we need to be trying to do is shortening this, overlapping. What's the minimum amount of information we need in phase 1 before we can go to phase 2, and so on. We have to have some preclinical studies. We have to have GMP manufacturing, but how can we do that more quickly? How can we get our phase 1 trial started more quickly, routinely, not just when there's a crisis, so that we can push this down and get through to licensure in under a year? This doesn't mean less testing. It means doing the testing faster. It doesn't mean missing out any of the steps that you would normally do in vaccine development. And in fact, in the SARS-CoV-2 trials, actually far more people were tested than is normal in vaccine development. So we're not missing anything out. We're just overlapping stages, cutting out the gaps so that we can go more quickly through all of this and get to the vaccines that we need to protect us against these different viruses that are still out there. And so one way we're thinking about it, because we don't know all of the viruses that might come and infect us, but viruses grouped into families. So SARS-CoV-2 is a coronavirus, Rift Valley fever is a bunyavirus. So if we have an example of each of these viral families that we do have a vaccine against, that we've successfully tested and is ready to use, if the next outbreak is caused by a new virus, all we need to know is which family does it fall into? Which of these viruses is it most like? Because then that tells us what technology is likely to be successful in developing a vaccine against that new virus. And at the moment, we don't have vaccines against these diseases. So if we had a new outbreak with something that was like CCHF, we wouldn't really be any better off because we don't have a vaccine against CCHF. So the aim is, we can't predict everything that's out there, but we know that they fall into these different categories. And by having a vaccine against one example of each of these families, we'll be very much better set up. These vaccines in themselves will be useful, but we can also use it as information to help us quickly develop new vaccines should there be a completely novel virus starting to cause an outbreak. And we do have many different vaccines in development already in the Pandemic Sciences Institute. But we're back to the very, very slow timelines that we were on pre-pandemic. In fact, things have gone even more slowly in recent times. So we're back to the very, very slow movement from one stage to the next and waiting for money so that we can complete this vaccine development. We hope to raise the money to get this done, but that's an uphill struggle at the moment. This is the work of very, very many people. We heard in the introduction about the importance of interdisciplinary working. This is work done by lots of different types of scientists and medics and nurses and volunteers and all the people who come and take part in the clinical trials, without whom we wouldn't know if the vaccines were effective or not. So I don't have a very detailed acknowledgement slide because there would be millions of people who need to be on it. So this is just to highlight some of the main groups of people: our trial team, the volunteers, AstraZeneca for their work in manufacturing the vaccine and also making it available without profit and in very large quantity, and our funders and the trial teams that we worked with across the world. So I'll stop there. Thank you.
got one word, and that's wow. I've got a billion questions, but it's not for me. So, look, this is now over to the audience, and we've got audience members online, and I see that questions are maybe coming to me, I'm not sure. And so I'm opening it to the floor, please. Who would like to ask the first question? There you go. 1 second, there's just a microphone. There we go. Our wonderful friends will run up and down the stairs.
Thank you for the brilliant presentation. I don't know whether you know, but Surrey has the highest COVID vaccination rate nationally. So you're virtually on the top. Well, I'm Jasmine. I was leading on the Surrey COVID vaccination program for the ethnic minorities, working with Surrey Heartlands and JCVI. It was a very difficult 17 months. I'm sure it was more difficult for you than what it was for us, trying to get the ethnic minorities vaccinated. And every time we tried making some progress, the media used to come up with some problem with the AstraZeneca vaccination, and everything used to stop. How did you deal with all these challenges? I mean, I used to often think about you and feel so sorry for you. So how did you manage to deal with all of that?
Well, again, it was teamwork, you know, and I think it was very important that it was teamwork because we did put quite a lot of effort into getting our message across to people. I did hear from some of the universities, eminent academics would just kind of give a response to the media kind of off the top of their head. And sometimes it didn't always come out in the most helpful way. But what we were doing in Oxford was getting together in a group and discussing what we were going to talk about in a press conference, rehearsing not only the facts that we were going to give, but the way we were going to present them, because they need to be accessible. We need to explain it in a way that people can understand, but we also must make sure that we don't accidentally oversimplify it and actually make people think we're saying something that we're not saying. And we should also be honest about when we don't know, because for a lot of the questions, the answer was, we don't know yet, but we're gathering more information, and we'll make those results available as soon as we have them. So we can't always know everything instantly, but we've always got plans in place as to how we're going to find out, but some of these things take time. So it was challenging. We discovered that in some cases there will be a completely spurious news report, which then other media outlets would report, because if somebody had said that the first volunteer in our clinical trial had died within days of being vaccinated, which of course was complete fabrication, then other media outlets reported that without bothering to check the story. So we had to ask the volunteer to do a video for the BBC with Fergus Walsh holding up a newspaper with today's date on it, with proof of life video. So no, I'm perfectly well, thank you. If you actually read the news story, it didn't make any sense, because I think at that time we'd vaccinated 6 people. Half of them would have received the new vaccine, half of them would have received the licensed meningitis B vaccine. And the news report said something like there were 4 people dead and 3 in hospital. Well, that's 1 more than we'd vaccinated, and half of them had the new vaccine anyway. So it was obviously complete rubbish. They weren't really trying to make any sense. They just wanted people to click on their news website. But then other people reported this headline that the first volunteers died. So we had to so we spent our weekend dealing with that, which was just complete fabrication. So it's hard to counter things like that. But we did do a lot of, trying to do information sessions, sometimes for ethnic minority groups, trying to find representatives who could talk to those people. I did webinars with a group of Ugandan bishops who wanted to hear it from where the vaccine had been developed, and then go and tell the people in their congregations what they'd heard from us. So yeah, quite a lot of work is needed to explain what we're doing, and also explain sometimes the boundaries of our knowledge at that present time.
Thank you so much for that. I think my last resort was I stopped using the term AstraZeneca, and I used to come and say, oh, why don't you come and take the University of Oxford vaccination? And that actually helped.
Really?
Because I used to say, oh, well, it's the world's best university, And they said, yes, of course we'll come and take it. Thank you.
Wonderful, thank you.
One question on funding. Has the government around the world taken seriously? This is a life-saving actually exercise you are doing, which I'm very thankful for, because my wife didn't want to take the other injection. Eventually she ended up taking AstraZeneca, and she's still there at home. So thank you very much. But my question is, funding, are the governments, WHO and all the national governments very aware that this need to be funded?
So we're not getting UK government funding at the moment. We have some philanthropic donations to support us, which we're extremely grateful for, but we are still looking for funding. In other countries, the government is supporting the establishment of new pandemic preparedness institutes. In Italy, for example, the government is supporting a new institute which will work alongside GSK, the company, as an independent research institute. But we're not seeing that here.
Wow. Can I, Al, please. Just here. There we go.
Thank you. Excuse my complete ignorance of the topic, but I'm very confused. You listed various viruses that we are worried about. You also identified areas in which they were endemic. So my question is, what are you waiting for? Why can't you go into those countries and deal with or attempt to immunize people there? Why you appear to be waiting for an outbreak? And I haven't understood.
Okay, so the countries on the map are where the viruses were first identified. It doesn't mean that's where they're going to be causing infections at any point in the future. So Rift Valley fever virus, first identified in Kenya, is now spread all across Africa and also into the Middle East. So it has a very wide distribution. It's not present all the time. We're not seeing cases in people all the time, but it's carried by insects. So mosquitoes, lots of different types of mosquitoes, can transmit the virus from one infected sheep to an uninfected sheep and cause an infection. And the eggs of some of those mosquitoes can remain dormant in the ground for up to 10 years. And then when it rains, those eggs will hatch and the grass will grow and the cattle come to graze and the mosquitoes are infected because they came from infected eggs, and they bite the livestock, and you will have an outbreak even though there's been no Rift Valley fever virus in that year for 5 or 10 years. So it's very hard to predict where it's going to crop up.
Okay, thank you.
Lady in the white blouse.
Hello, thank you for the lecture and for your work. So I've just started a PhD at the Health Protection Research Unit, one of them, focusing on adolescent vaccine hesitancy. And I wondered what you think we as the scientific community can do to raise trust in vaccines and in science going forward.
Well, what I find really difficult to deal with is the fact that there are quite a lot of people who don't get their news from traditional outlets, and they get their news from places that will just tell fabricated stories, and these get amplified and passed around, and people seem to believe they're true without any evidence behind them. So it's very difficult to intervene in those circumstances, and it's hard to get people to trust that the information they're being given from a traditional news outlet when they're getting information from their friends that's telling them something completely different. So I think I really don't know how we deal with that. I mean, having ambassadors, people who can engage with those networks and can actually give the true response. We have websites. In Oxford, there's a website called the Vaccine Knowledge Project. It puts out information about, it started with all pediatric vaccines that are used in childhood, but then there was a special section of it talking about the COVID vaccine, just explaining the facts, what's in the vaccine, how does it work, who should have the vaccine, all of those things. And it's medically checked and correct information, but written in a way that's easy for anybody to understand. So the information's there if they want to go and find it. It's difficult to persuade people sometimes they should go and find that information.
I think that's an excellent question, actually. And if I can just add, so funders such as the Wellcome Trust or UKRI, Medical Research Council and things like that, they put a big push on the public engagement of science, but they need to do more. And it was, I think historically it was very little. It's like if we spend too much money on that, we're not doing enough money on the science. But if you can't drive that impact and that true knowledge, which is what Sarah's talking about, then we're in trouble. So that public engagement will drive that trust, but it has to be driven by the right stakeholders, i.e. the brains behind it and then the mechanism to disseminate it. The problem is the internet is full of incredible things. That's polite. I'm just going to look at this side of the room. Sir.
Thanks for your work. My question is about your timeline, your slide, which has a year of lead time to live, effectively. And while sort of greatly admiring the platform technology that you've got, that sort of shrinks the sort of release of that initial batch to the left, I'm just wondering if what we could do to shift acceptance to the left as well, both in terms of the clinical trials and then the public acceptance. Is there ever going to be a day where we say that the same platform virus has the same side effects? So we don't need to test that. We know it's safe. Is there ever a day where we take the general population and say, you will always have a vaccine? You will know that it's the same platform. It's just that we'll tweak at the last minute what goes into it. So the whole thing could shift to the left by half, and it just will increase the acceptance both at a regulatory level, a consumer level, and dare I say a funding level as well, because it'll be funding the whole platform rather than an individual response to an individual virus.
So there are definitely ways we can speed up that timeline. As I said, we haven't ever planned to go fast. We hadn't practiced it. That's the best we could do in the circumstances, and we could definitely, we did things in the manufacturing that didn't work first time, and then we had to stop and go back and do it again. So we got our first clinical trial started by April, 23rd of April. I think we could have at least shave a month off that timeline, going through normal regulatory processes. Actually, the regulatory processes didn't slow us down, because what the regulators did was, as soon as we generated any piece of information that they would normally want to see, we sent it to them, they reviewed it, and that meant that by the time we had the last piece of information, they reviewed it in hours. Whereas normally, you accumulate a big dossier of all the information, and then you hand it over, and it takes weeks, because they've got to go through the whole lot. They didn't do that this time, so they helped us speed up. There are ways we can speed that up. There's the testing of the vaccine. We could use more modern methods for that. So it takes us 3 weeks to say that there's no bacteria in this vial of vaccine that's come from this lot, because that's the approved test. We've got molecular sequencing methods now. We should be able to do that much more quickly, but somebody has to spend the money. I mean, in principle, we know how to do it, but somebody has to spend the money to prove that it always works. So if we're going to switch to a very rapid test to say there's no bacteria in the vaccine, we have to be absolutely certain it's going to work if we're then going to use that and start injecting people with it. So there's quite a lot of work which will take time and a lot of money to achieve that. In the meantime, we're stuck with it takes 3 weeks to do the test before we can use any of the batch. We need efficacy testing, though. So we immunise a lot of people, and we have to wait and see some of them get infected before we know that the vaccine's working. Now, in a rapidly spreading outbreak, like when the Ebola cases were increasing very rapidly in West Africa in 2014, you don't need very many people because the rate of infection is so high. But in the UK in 2020, of course, we kept going to lockdown. And so every time people stayed at home, then they weren't having any possibility of getting infected, so that we couldn't tell if the vaccine was working or not. We needed trials around the world where the virus was moving around at slightly different rates, but it still took time to accumulate the data. So if for viruses we already know about, we can test them, we can have stockpiles ready to go, so that there's an outbreak somewhere, we've already got a stockpile, and we've got the clinical trial protocol that's agreed on, and we just get on and test it as quickly as possible. That's how to accelerate. For the unknown viruses, we have to have our manufacturing methods tested, ready to go at speed. We have to have our clinical trial methods tested and ready to go at speed. But then we need the vaccine manufacturing facilities to make large numbers of doses, because by the end of 2020, we knew that some of the vaccines were definitely effective and should be used, but there weren't enough of them to go around because it takes time to ramp up the manufacturing. So that's the other part of the piece. It's no good knowing that a vaccine is highly effective, but you haven't got very much of it when the whole world needs it. We've got to be prepared with the manufacturing facilities as well. And those manufacturing facilities can't just be set up and then sit there waiting for something to happen, because then they'll take too long to get going again. So the next piece of the puzzle is, what do those manufacturing facilities do in the meantime while we're waiting until they're needed again? So more things to think about.
Multifactorial.
Yep.
Lady in the blue shirt.
Thank you. And thank you very much for your talk this evening. I brought my daughters along with me to hear about the amazing work that you've done, how hard it can be and challenging at times, but also how fulfilling and impactful the work is particularly in science. And I just wondered if you had any words of advice or inspiration for young people going into these careers. You know, what have been your highlights? What do you love about it? What advice would you give them?
So I really like working out how things work and then how you can use that information to do something and make a change. So that's always, you know, since I was at school, that's what I wanted to know. Understand how the body works, understand how we can do things to help either protect against disease or cure disease, and there's lots of different ways of doing that. So I think what you need to do is find something that you're really interested in, that you want to do, and then carry on doing it. And in vaccinology, it's a very, very broad discipline. So we have nurses, we have play assistants, whose job it is to entertain children who are taking part in vaccine trials so they don't get upset by having a blood sample taken. We have, you know, people who are very people-orientated, but at the other end of the scale, we have people who spend their entire day sitting in front of a computer working on very large data sets and pulling the relevant bits of information out of those data sets and modelling the data and using, you know, mathematical techniques to understand what's going on when we're doing these vaccine trials. And in between, there are people working on vaccine manufacturing. They have to be very, very precise. When you're manufacturing, everything has to be absolutely perfect and exactly the right way. There's no room for sloppiness. So you need a really dedicated type of person to do that sort of work. But then you also need people who are willing to go out and talk to volunteers about taking part in clinical trials. Lots and lots of different skills. So I think work out which bit of science it is that you like, and then try and follow a career where you can follow that interest.
Thank you.
Sir. Just here.
The pandemic seems to have dropped out of the media spotlight quite a lot, but is it over? What's happening to infection rates around the world? Is that a fair question?
So this virus has now moved into a territory that's not my area anymore. So I work on the bit at the beginning when it's an outbreak and we have to respond. But what I can tell you is that, as I said, there are other coronaviruses that regularly cause infections. In the UK and around the world, there's 4 of them. They're very non-memorable names. You've probably never heard of any of them. And we don't even diagnose them unless there's an outbreak where, say, people in a hospital or a care home all start to have a respiratory illness and be very ill. Then they might be tested and find out actually it's a coronavirus that's causing this. And then all you can really do is isolate them and stop anybody else getting it. So those viruses, we get infected in childhood. We'll have a cold, not be seriously ill. Everybody knows, be off colour but not be seriously ill, and we start to develop some immunity, and then we'll get infected again later on, but it'll just be a mild illness. We don't get lifelong immunity. We continue to be infected throughout life, but the only problem is when a brand new coronavirus that we haven't seen before in childhood, we haven't had the opportunity to build up immunity to, when that starts to circulate and nobody has any immunity to it, it spreads rapidly, causes lots of disease. That's when we have the outbreak or the pandemic. So what I think is going to happen is that SARS-CoV-2 is going to start behaving like these other seasonal coronaviruses, which are already adapted to infecting people. So SARS-CoV-2 came out of bats. It's going to go through a lot of adaptation as it starts to infect people and spread through people. It's going to mutate, and the things that work best for a virus that infects people will be the things that persist in that virus. I think that rate of change is going to slow down now. It's very well adapted to people. I don't think it's going to be changing as quickly. It will still cause infections, as the other coronaviruses cause infections, but I don't think it's going to cause us so much of a problem.
I think that's a really good point, because you stated, I think we are constantly under attack. You know, there are labs around the world. We've got a lab not too far from here called the Pirbright Institute, which are monitoring these kind of infections around the world. So we talk about this pandemic, it's multi-pandemics all the time, so we have to be aware of that. Hence your question, sir, about trying to make it faster. It's goes back to that one point, and what's that one point? Funding.
Yeah.
Did you pick that up? I'm going to go towards the back of the room. There we are, right at the very back.
I've got some acquaintances who are now saying that the post-pandemic complications, diabetes, heart disease, things like that, are more related to the vaccine than the virus. How would you counter that rather than just saying you're wrong, which is my scientific limit?
So just, correlation is not causation. Just because one thing happens and then another thing happens afterwards, it doesn't mean that the first thing caused the second thing to happen. So that's why we have the controls in the clinical trials. That's why we have half the people giving the novel SARS-CoV-2 vaccine, half the people giving a different vaccine. And it's actually amazing when you record people's, what we call them adverse events after vaccination, but it can be very mild things. So we ask them, do you have a headache? Do you have a sore arm? Do you have a raised temperature, or do you feel feverish even though you haven't got a raised temperature? All of this is recorded in great detail in the early phase clinical trials. And if you do that with people that you've given a saline placebo to, you will record quite a lot of people feel tired, people have a headache. It's not necessarily due to, they've got a sore arm because a needle's gone into it. It's not necessarily due to the vaccine itself. It's due to, sometimes to the emotional upset of going and getting vaccinated and having to be asked all these questions, and you feel a bit tired at the end of it. But that's not necessarily down to the vaccine. So it's really important that we have controlled trials where we are looking at what happens in the people who didn't have the vaccine as well as the people who did. So then we can start to see what's, in that case, when we're given two groups of people who are matched, either not the vaccine or the vaccine at the same time, we can compare and see what is actually likely to be down to the vaccine. For longer term effects, that becomes very difficult because eventually everybody got vaccinated. But vaccines don't cause late effects. Vaccines have a rapid response in the body. They stimulate an immune response, and that can make, in itself, that can make you feel quite ill. That can make you feverish in the first 24 hours, 48 hours maybe after vaccination. The fact that your immune system's being fired up can actually make you feel quite unwell. That dies down, and we rarely see anything happening as a result of vaccination after the first few days. Long-term effects, there's no explanation for why they could be caused by vaccination.
Middle of this block. I'm trying to go around it, sir.
That was great, that lecture. Thank you very much. I'm former AstraZeneca. Okay. I left the company 10 years ago, and I was responsible for putting in new facilities all around the world, not for viral, for oral solid dosage and things like that. And we had McKinsey look at us and say, you guys know what you're doing, and walk away. We were not building factories as quickly as you did. All right. How on earth did you manage to put in such an incredible track record like that to get those facilities up and running and producing vaccines of that scale?
So those weren't new facilities. Those were AstraZeneca reaching out to other manufacturers and asking, do you have a facility that's ready to go? Do you have everything? Do you have the staff? Do you have the capability to take the technology from us and go very, very fast? And so in Brazil, for example, there's a company called Fiocruz. It's part government owned. It makes a lot of other vaccines. It's never made a viral vector vaccine before. But they are a vaccine manufacturing company, and they received the instructions and they received the materials of how to make the vaccine from AstraZeneca. To begin with, they were just receiving vaccine that was made in bulk elsewhere, and they were doing the putting it into vials, so the last stage of the manufacture. And that could be, those vials were then being used in Brazil and South America more generally. But then they took on the whole manufacturing process, and so they managed to manufacture tens of millions of doses of vaccine, which were then used in South America. The problem in Africa is that there were not facilities that were ready to go. There were some manufacturing facilities, but they weren't modern enough, or they weren't large enough. They didn't have the staff that could take on a new technology very quickly. That network of manufacturers was many, many different companies all working as one, but the facilities have to be there before we started to work with them.
Okay, thank you very much. Excellent.
So from your data, I mean, a lot of the origins of these viruses are in Africa, from what you
Yep.
see, but the manufacturing facilities are not on the continent.
Yeah.
So this is something clearly that I'm hoping that the WHO and the World Bank are looking at and thinking, we do need to put a site readiness,
Yeah.
or indeed it would be used a lot of the time anyway. It's a no-brainer to me.
Yeah, and this is the aspiration is that that will happen. So there's a manufacturing facility in Dakar in Senegal, for example, in West Africa, which makes a lot of the world's yellow fever vaccine, but it doesn't make very many other vaccines. That's now being expanded, so that will have new facilities, new capabilities, expand the staff and be ready to produce more vaccines. Kenya has an aspiration to start producing a lot of the vaccines, the routine childhood vaccines that are used in the country. They want to produce quite a high percentage of them within the country.
Right.
The difficulty is that those vaccines are likely to be more expensive than the ones that you could buy cheaper from the very, very large manufacturers that produce them in huge quantities and then ship them into Africa. And so the governments of those countries are going to have to support, if they want vaccine sovereignty, if they want to be able to manufacture locally for the local population, which is the best way of ensuring that population will have access, then the governments will need to support them by keeping those facilities running and buying vaccines from them, even though it would actually be cheaper to buy them from the existing bulk manufacturers. So we don't know how that's going to work out at the moment.
Yeah, I understand. Sir, you just turned around.
Thank you. You put up a slide showing a number of diseases where we currently don't have vaccines developed, where you quite rightly suggest that we should develop them, stick them in the freezer so that we're ready. And so that actually shows that we still have a burning platform, even though it doesn't feel like it with COVID. You know, these things are all out there ready to attack us. So can you give us an idea of the scale of the funding need? What does it cost to move from where the technology now is to develop a vaccine for one of these that is fully tested and approved? What sort of scale of funding are you looking at?
Well, it depends how far we're going. So, you know, getting into a phase 1 trial is maybe, if it's an academic-led clinical trial, we're very cost-effective. That might be a couple of £1 million to do the manufacturing in a small phase 1 trial. Then you sort of multiply that by at least 10 if you want to do a phase 2 trial. If you're going to do an efficacy trial, if you have the opportunity to do an efficacy trial, then, you know, probably multiply by 10 again. But in many of these cases, we don't have the opportunity. So what's going to happen is that, you know, we spend £5 million on making a stockpile and having it ready. And then if there's an outbreak, you use your stockpile, and then there'll be some costs involved in doing the testing of those stockpile. So yes, there certainly are costs involved, but compared to what happens, you know, what gets wiped off the economy when there's even an outbreak in the country, as happened in South Korea with MERS, it's really much more cost effective to spend the money up front and get these vaccines ready.
Wonderful. Right, I've got time for one very final question, and I don't know where to go. I'm going to go to Vib.
There has been a lot of discussion among ordinary people about the two different types of vaccines, generic types in terms of Oxford and Pfizer. One of them, I guess, is a new type of approach to creating a vaccine, if I understand it right. What does it say for the future of making vaccines and the efficacy preferences between the two systems?
So when the vaccines are compared head-to-head in the population as opposed to in the efficacy trials, there's not very much difference between them. Also, the Chinese vaccines, which is another old technology, inactivated vaccines in Hong Kong, when they were compared head-to-head with the mRNA vaccines for the Omicron outbreak, they worked just as well. I think the mRNA vaccines are a really exciting technology. They'd hardly been used at all prior to 2020, and obviously there's been a massive expansion in the use. They will be used against some of these other outbreak diseases as well. There are some issues still to solve, but I don't think that we should expect that the development of that technology will stop. So we need better thermostability. Instead of having to store them at minus 80 degrees, we want to be able to store them in a fridge. How can we change the way the vaccine's made to achieve that? Because that would be a very beneficial thing to do. There are some adverse events after those vaccinations. They give you quite a strong reaction. How can we change that and still get the strong immune response? So there's more work to do, but I think that they will be used. They can't be used against every disease. So for some bacterial diseases in particular, like pneumococcus, where we have the glycoprotein conjugate vaccines, mRNA vaccines can't replace those, but they can be used against many different diseases. They're being looked at for use in cancer vaccines as well.
Great question. So thank you very much. But before we do a round of applause, I'd like to offer you a gesture of our thanks. It's a little memento.
Oh,
there you go.
thank you very much.
There you go. I have no
Thank
idea
you.
where you will put that.
Okay.
Thank you.
Thank you.
So, ladies and gentlemen, please. Stay seated.